What a beautiful day. My phospholipid bilayer is feeling awfully fluid. Oh!
Plasma membrane.
Sorry.
Stop and check the intruder.
Stop right there.
I'm sorry, I'm late for an interview.
I'm just checking.
Why is that necessary? I'm a ribosome, just the same as any other.
My job is to allow only certain molecules to enter the cell. Sorry, we're trying to be very selective.
Look at this guy. You can't even remember that his transport proteins help materials move across him every day.
My receptor proteins need to bind with substances outside the cell. And my transport proteins need to assist the transportation of materials across me every day. Do we still have a problem?
No, sir. I apologize. I really need this job, though. I haven't had RNA in a long time, and my protein-making skills are going to waste.
Put your arms to the side. I have to pat you down. All right, you're clear to go.
Thank you.
Ribosome, you're late.
I know, sir. I apologize. The cell membrane needed to check me to make sure I wasn't harmful to the cell.
Well, that's good. As you can see, he is one of our best employees. I do direct him to be very picky, like I direct most cell activity.
I can tell. That side carbon chain scared the proteins out of me.
Well, have a seat.
Okay. And sorry to interrupt, but what was all that jelly-like liquid outside of your office?
Oh, that was my nuclear envelope. It stores most of my contents, similar to my friend Mr. Cytoplasm. First of all, I have to do a background check on you to make sure you're not a 50S, 30S prokaryotic spy.
Of course, sir. I understand. No need to worry. I am a 60S, 40S ribosome, just like any other in the eukaryotic cell. My large substrate is known as 60S, and my small substrate is known as 40S. I was composed of 4R RNA, or ribosomal RNA molecules, and a complex protein, unlike prokaryotic ribosomes, who are only composed of 3R RNA molecules.
So, Mr. Ribosome, why do you feel like you're a good fit for this cell?
Well, I'm very crafty when it comes to making proteins. I take mRNA, or messenger RNA, and put it in between my two substrates. I am a very welcoming ribosome. All the mRNA love me. From there, I use mRNA to gather tRNA, or transfer RNA, which then attach amino acids to me, forming a large chain. From there, I am able to make proteins.
That's good to know, because in my nucleolus, there's constantly RNA being made. It also might serve you well to know that my nucleolus also stores much of the cell's DNA. By the way, your substrates do look pretty cozy.
Not to be rude, sir, but you have me feeling pretty awkward.
Well, you shouldn't be, because you've just been hired.
Thank you so much.
No problem, Mr. Ribosome.
I promise I will give you my full effort.
You better, or I'll have the plasma membrane throw you out of one of his pores.
I just got the job. Time to go to work. Our cell just got an organ now.
A very important one.
He makes proteins from RNA.
It's me, the ribosome.
But don't forget the security guard.
He keeps the cell safe.
He controls what comes in and out.
It's me, the plasma membrane.
Last but not least, there is the boss.
He directs the cell activity.
It has a nucleus with DNA.
It is me, the nucleus.
Yeah!